SIV-induced translocation of bacterial products in the liver mobilizes myeloid, dendritic, and natural killer cells associated with liver damage.
All right, so I guess first I should say thank you to all of you for hanging in there till the end. I promise I will be quick. And second, I'd like to thank the organizers for giving me the opportunity to share with you some of our recent data on the relationship between microbial translocation, innate immune activation, and subsequent liver damage. Okay. So first off, it's becoming more and more clear that one of the major complications of HIV infection is in fact liver disease. And so some of the things that I'm going to talk about is what are the underlying causes of what's causing this liver dysfunction in HIV. So one of the first, of course, that we haven't really dealt with necessarily in the past are drug toxicity due to the various antiretrovirals that HIV-infected patients are taking, as well as other drugs that they may be administered just to control other symptoms. The second of these, of course, is co-infections with various hepatitis viruses, hepatitis B, hep C, as well as other hepatovirus co-infections. And then the third possibility for liver disease and HIV infection is what most of my talk will focus on today. And those are complications that are not due to drug toxicity, not due to an underlying co-infection, but are actually dealing with the virus itself. And that can include non-alcoholic fatty liver disease, NASH, as well as hepatocellular carcinoma, which are much more common on HIV-infected patients than those uninfected controls. And so since we are talking about, for the purposes of my talk, comparing to SIV-infected macaques, I can just show these slides briefly, that if you look in the livers of SIV-negative compared to SIV-infected macaques, both by H&E stain, you'll see up in the top panels, and by trichrome at the bottom, is that SIV-negative livers look nice and healthy, not too much going on here. But in animals that are SIV-positive and are progressing to AIDS, you see a variety of symptoms that are indicative of hepatitis. Lymphocyte infiltrations, fibrosis, as we've talked about earlier today, a variety of cell necrosis, and just generalized inflammation in the liver of these infected animals. Indeed, in addition to seeing liver pathology, we can also look in SIV-infected macaques to look for the surrogate markers of liver complications. And again, we'll see the same thing, is that these common markers in the sera, alkaline phosphatase, aspartate transaminase, we'll see are both significantly increased in the serum of SIV-infected compared to uninfected animals. And we will see a little bit of an increase in alanine transaminase, which is again a common marker used in humans, but it's not significantly upregulated in this cohort. All right. So just to switch gears briefly, a topic that we've spent a lot of effort going through today, and that's the phenomenon of microbial translocation as a primary mechanism of immune activation systemically in HIV infection. So the fundamental premise, as we've discussed thoroughly today, of microbial translocation is a breakdown of the gastrointestinal tract, where microbes and microbial products move out of the lumen of the GI tract and into the circulation. And this serves as a potent source of systemic immune activation. So one thing that's very important to consider here is that I think most of the research focusing on microbial translocation has looked extensively on what's going on, what are the mechanisms of dysregulating the gut epithelia, and then what are the mechanisms of actual immune activation once these have moved into the circulation. But I think one thing that's been overlooked to a large degree is what goes on in between. And so that's what brought us back to our discussion of issues with the liver, is that roughly 70% of all the venous blood that's coming out of the intestinal tract is actually going through the liver before it moves into the circulation. So this really puts the liver right in the middle of all these microbial products that are coming out of the GI tract. And so that's what we'll be focusing on now. So in fact, if you look in a cohort of SIV-infected macaques, what you'll see is this is just staining for LPS by immunohistochemistry. SIV-negative animals, you see very little LPS. But if you look at an animal that's progressed to AIDS, you actually just see a large a large volume of LPS staining positive cells here, and you can see this even corresponds with some lymphocyte infiltrates. And then if you look at the total increases in liver E. coli as well as LPS, you see significant increases in SIV infection of both of these. And again, this just speaks back to the fact that the liver is a major filtering organ, and you're getting an accumulation of microbial products here in addition to those that have translocated into the circulation. So I think this led us to ask the question that in addition to systemic activation, is this a potential source of local activation in the liver? And so I won't spend too much time on this. Most of you are familiar with pathogen-associated molecular patterns. We've talked about it today. Primarily, we'll focus on LPS, just because it's signaling through TLR6, TLR2, and primarily TLR4. And this is the one that we'll be most concerned with today of what's going on in the liver. So one of the major cell types that we find in the liver that we're particularly interested in is myeloid dendritic cells. This speaks basically to the fact because they express large quantities of intracellular TLR4, which can bind LPS. So this just gives you a flow cytometric plot of how we identify myeloid dendritic cells. CD45 positive, live cells, negative for CD3, HLA-DR positive, negative for CD20 and 14, but positive for CD11C. And in fact, if we look in the livers of SIV-infected macaques, we will see a significant increase in the total frequency of myeloid dendritic cells. But I think perhaps more importantly is that if we look at the functionality of myeloid dendritic cells in the liver of infected macaques, for one, we find that most of the CXCL16 positive, which is a major liver chemoattractant, cells in the liver are in fact MDCs. And that if you look at the total frequency of CXCL16 positive MDCs in SIV-infected macaques, it is elevated, and that even if you go back into an in vitro experiment, you take naive MDCs, you stimulate them with LPS, you induce a large production of CXCL16. And so just to sort of summarize this, MDCs, major sensors of LPS in the liver, they're major producers of CXCL16, which is a major liver chemoattractant, and they produce CXCL16 specifically in response to LPS. And just to do some correlative analysis, if that we look at the total frequency of MDCs in the liver, you see a very nice correlation here with the frequency of MDCs and the total area of LPS as we detected by immunohistochemistry. And you see the same correlation with the frequency of liver E. coli. And again, these correlations hold up for the most part for CXCL16 positive MDCs with LPS and E. coli. And what this suggests is that there really is a direct relationship with the frequency of MDCs as well as those that are producing CXCL16 in the liver. So this implicates that they could have a major role in recruiting CXCL16 binding cells to the liver. And so that's actually the next thing that we investigated is what might be occurring on the CXCL16 and its receptor CXCR6 axis. So the first thing we did is we looked at just major lymphocyte subpopulations in the circulation of naive and SIV-infected macaques. And indeed, that's exactly what we see, is that on natural killer cells, we see a significant upregulation of CXCR6, or at least an expansion of the CXCR6 positive population. A modest increase in CD8 T cells, although this was not significant, and then also an increase in CXCR6 positive, CD4 positive T cells. And what this indicates to us is that in instances of infection where we have potential increases in CXCL16, we are also mobilizing the CXCR6 potential liver trafficking cells in the circulation. And so for the rest of the talk, we'll focus primarily just on NK cells, and I think it's important to point out as we're going forward is that there is a very unique feature of CXCR6 positive NK cells as well as other CXCR6 positive lymphocytes, and that's that they tend to be hyperfunctional. And you can actually see this here just in a standard 4-function ICS assay, is that CXCR6 positive NK cells, both those from infected animals and from naive animals, are hyperfunctional compared to their CXCR6 negative counterparts. They have higher CD107A as a surrogate marker for cytotoxicity, increased interferon gamma and TNF alpha production, as well as increased production of MIP1 beta. Okay. So since we're looking at NK cells and potentially we have a mechanism of increased NK cell trafficking into the liver, what's actually going on with NKs that are actually already in the liver? So one thing that we noticed initially is that the bulk NK cell population in infected animals, the bulk in infected animals is actually increased, and then we see a stability of the CD16 positive population. Okay. But this corresponds to something else that's going on here, is that we also have an increase in the apoptotic NK cells, but no changes in Ki67. So that seems a little bit disjointed. But what we actually see, or what we actually interpret from this, is that even though there's increased apoptosis, there seems to be stability of the CD16 positive cytotoxic NK cells, and we think this might be compensated by trafficking of the CXCR6 positive cytotoxic NKs from distal sites. And so finally, just to address the functionality of the hepatic NK cells, is that they are indeed in the liver becoming more cytotoxic. We see increases in perforin, we see increases in CD107A, but in general, very little change in cytokine production. Okay. So just to sort of pull all of this together, one thing that's important to point out is that we know liver damage, at least in these cohorts, does not seem to correlate with viral load. But if we look at other measures in this study, we see ALP and ALT, they both correlate positively with the percentage of liver E. coli, percentage of liver area LPS. They also positively correlate with the infiltration of MDCs and cytotoxic CD16 positive NK cells into the liver. So what this really tells us is that even though liver damage may not be specifically associated with replicating virus, it is positively associated with this increase in microbial translocatants in the liver and this subsequent infiltration of innate immune cells into the liver in response to the microbial products. So just a real quick summary, microbial products translocating from the GI tract accumulate in the liver, not surprisingly because it's filtering venous blood coming from the GI tract. Microbial products in the liver induce MDC and NK cell mobilization and enhanced activation. And overall, we see an increased innate immune activation associated with surrogate markers of liver damage. And I think kind of the take home here is that it's just another facet of the microbial translocation story, is that there may be a potential link between microbial translocation and complications of liver disease in HIV and SIV. So with that, I'll just thank a few people from my lab, Haiying Li, Jamie Schaefer in particular, also a lot of the staff at the New England, now closed New England Primate Center. Also definitely Jake Estes for the beautiful immunohistochemistry, Jason for help with samples, and of course all the funders. And with that, I'll close and take any questions. Crystal clear.
So thank you very much. I think we finished in time, only 2 minutes late, and I would like to thank all the presenters and everybody in this room for coming and being interested in this hot topic in our research. And thank you, Jason, again for introduction. Thank you. One question for you, which my main question is about this microbial. So, thank you. Thank you very much. Thank you. Yeah. So it's clear that from what you told me, all these people, and what we know from this microbial change of HIV. So it is a kind of defense mechanism or part of the pathogenesis. Should we aim to restore it or help to keep abnormal?
Right. So your microbiome things the way that it's balanced.
But is things like decrease, let's say, temperature with high fever? Or lymphocytes in
So
the
it becomes a cycle. So the proteobacteria are more close. So they have no T cells, they have no so they do a better job of getting in. And then they cause it. Many of the things are bypassed cause that.
But there is no defense aspect of this.
No, and in fact, your microbiome has its own defense against proteobacteria. So